Welcome to science class. Today we are going to take a look at mitosis, the division of cells in multicellular eukaryotes. We touched on this last time, but you take the average human cell, which is a tenth the size of a millimeter, and if that one cell divides into two, those two divide into four, and so on, that process only needs to occur 45 times to produce approximately the number of cells in a full-grown adult. This is not how it actually works. You reach the 32-cell morula stage at around 96 hours or 4 days. At that rate, you'd be adult-sized in only 36 days. After 9 months, you'd be the size of the sun. We are going to cover development in an upcoming video. For this video, we're going to discuss what happens leading up to and during cell division. Let's get started. Mitosis is the division of one eukaryotic cell into two identical daughter cells. Rather than talking about mitosis in terms of the cell dividing, we often say the nucleus divides. The goal is that mitosis produces two genetically identical daughter cells. In order for that to happen, the cell must first make a copy of its entire genome and then evenly split during mitosis. The importance of that can't be overstated. Let's back up a bit and start from the very beginning. Actually, first we have to talk about DNA. A cell contains a ridiculously large amount of DNA. A typical human cell, a tenth of a millimeter wide, contains a combined total of 1 meter of DNA, 10,000 times the length of the actual cell. DNA strands are only a few nanometers wide, so they easily fit inside of the cell, like how a mile's worth of fishing line could easily fit inside of a backpack. The DNA in your cells is separated into fragments of DNA, which are bonded with protein structures called histones. DNA mixed with histone proteins is what we call chromatin. Now imagine that mile's worth of fishing line in your backpack. Say you had to transport it to another backpack. You really don't want to comb through miles worth of fishing line. That would take forever. Instead, you want it to be packaged. Our chromatin does this by compressing and condensing itself into chromosomes. Chromosomes only form during mitosis. Otherwise, the DNA is much more loosely stored, so that all of it is accessible. Humans have 46 chromosomes, and you inherit 23 from each parent. Those chromosomes are complementary. They each have the same genes, apart from the sex chromosomes in men. For every chromosome you got from your dad, there is an identical chromosome you got from your mom, except for the Y chromosome in males. Each complementary chromosome will have different versions of each gene, but they're still the same genes. These complementary chromosomes, known as homologous chromosomes, look a little something like this. The little strand they form is called a chromatid. This will all become very important shortly, so keep all of that in mind. Let's say we have two cells that have just finished dividing. We'll pick one of them. This cell is in a portion of its life cycle, which we also call the cell cycle, known as interphase. Interphase is the part of the cell cycle where the cell is not dividing, but just growing and going about its normal cellular activities. The first stage in interphase is the G1 phase, or first gap phase. There's nothing much to talk about. The cell grows larger, it makes some new organelles, and goes about its business. The G1 phase typically is the longest phase by a healthy margin. The next phase is the most important, and it's the S phase, or synthesis phase. During the S phase, the cell duplicates all of its chromosomes, and by extension, all of its DNA. Let's go back and look at those chromatids we mentioned earlier. After DNA replication, each chromosome contains two sister chromatids. This is part of how the cell ensures each daughter cell it produces is genetically identical. These chromosomes break in half, and each sister chromatid goes either to one cell or to the other. The chromatids we saw earlier, those actually only look like that way at the end of mitosis. During mitosis, the chromosomes form these X-shaped duplicated chromatids. Again, during the S phase, all of the DNA is duplicated. but the DNA does not form chromosomes. It's still chromatin at this point. The confusing thing here is that chromatid and chromosome are interchangeable. This is a chromosome composed of one chromatid. This is also a single chromosome, but composed of two identical sister chromatids. The final phase of interphase is the G2 phase, or second gap phase. This is generally the shortest phase in the cell cycle, and all that really needs to be said about it is that the cell gets ready for cell division. Some proteins that help that process are made, some enzymes get produced to kickstart that process, but that's really it. Next in the cell cycle comes the M phase, where mitosis and cytokinesis take place. We're not going to go into super deep detail on each step in mitosis. Your teacher will do that for you. Like with photosynthesis and cell respiration, I just want to show you the process from beginning to end. The M phase begins with the 4 stages of mitosis. First is prophase. Prophase is the longest and most complicated stage by far. One major event that takes place is the complete breakdown of the nucleus. In order to split the sister chromatids, they have to be accessible, so the nucleus needs to go. So at the beginning of prophase, there's a nucleus, and by the end, it's all gone. At the beginning of prophase, there are no chromosomes either. Chromatin gradually condenses and coils itself into chromosomes throughout prophase. Finally, a structure called the mitotic spindle forms. The spindle is a collection of cytoskeleton filaments, which are going to be used to attach and then pull the chromatids apart. There are actually 2 spindles for pulling the chromatids to opposite sides. Think of lassoing something, then pulling the rope towards you. That's kind of what the spindle does. The next stage is metaphase. Some sources distinguish a phase between prophase and metaphase called prometaphase, but most high school books don't. 2 important things happen during metaphase. The first thing is that the 2 spindle apparatuses connect to each chromosome on both sides. The region on the chromosome where the spindle attaches is known as the centromere, but what the spindle actually attaches to is a structure called a kinetochore. The other step in metaphase is that all of the chromosomes line up in the center of the cell. This helps organize the chromosomes so that they are evenly split on both sides, giving each daughter cell the same number of chromatids. Next comes anaphase. This is the simplest stage in mitosis. The spindle fibers, which are attached to the kinetochores on each chromosome, shorten and pull the sister chromatids apart. The moment the chromatids separate, metaphase ends and anaphase begins. But anaphase continues until the spindle detaches, meaning you can find a cell at various different points during metaphase. The last step in mitosis is telophase. Telophase is the exact opposite of prophase. The nucleus begins to reform, the spindle breaks down completely, and the chromosomes unwind back into chromatin. That completes mitosis, but it does not complete the M phase of the cell cycle. Technically, this is still just one cell. It just has 2 nuclei. I can go from one side to the other without passing through a cell membrane. Cytokinesis is the final step of the M phase and the final step in the cell cycle. Cytokinesis is the splitting of the cytoplasm between the 2 halves of the cell, thus creating 2 daughter cells. In animals, cytoskeleton structures pull on the cell membrane from within to pinch the cytoplasm in half. In plant cells, a cell plate begins to form between the nuclei, which eventually separates the cytoplasm in each half with a fully formed cell wall. Some cells divide throughout your entire life, such as your skin and the lining of your gut. Other cells, like skeletal muscle and nerve tissue, don't divide. They only grow larger. A bodybuilder does not have more muscle cells than you or I do. They simply have much larger cells. These cells are stuck in what's known as a G0 phase, which is a bit like an extended G1 phase for our purposes. And that is the cell cycle, more or less. Next time, we are going to look at how the cell cycle is regulated. How does a cut heal so well? How does a cell know when it's time to enter the G2 phase? What about when a cell loses the ability to control its growth? Those answers and more will be in the next video. Thanks for watching.